Today's Monday, December 14th, 2015. I'm Joe McMaster. As part of the MIT Infinite History Project, we're talking with Dr. JoAnne Stubbe, the Novartis Professor of Chemistry and Biology, who's been a faculty member at MIT since 1987. Her work as a biochemist over the last 4 decades has had profound effects on fields ranging from the synthesis of biodegradable plastics to cancer drug development, and her groundbreaking research in the field of enzymology has advanced our understanding of the chemistry at the very root of life. Professor Stubbe has been praised as a scientist's scientist and a passionate teacher, and in 2011 she received MIT's James R. Killian Award for Faculty Achievement. Numerous national and international awards celebrating her achievements include the National Medal of Science, the Welch Award in Chemistry, the National Academy of Science Award in Chemistry, and many others. Professor Stubbe earned her Bachelor of Science degree in Chemistry at the University of Pennsylvania and a doctorate in Organic Chemistry at the University of California at Berkeley. Well, thanks for talking with us. So let's begin with your work. Maybe you can tell us about enzymology and enzymes, and just first of all what they are, but also what this has been the center of your work, as I understand it. So maybe you can tell us a little about that.
So enzymes are proteins, mostly proteins. There are a few RNA, ribonucleic acid enzymes, and what they do is they accelerate the rate of a reaction. If you're converting some substrate to some product, they can increase the rate of the reaction by 10 to the 15th. So everything that goes on inside our body, how you break down sugars, how you break down fatty acids, how you make sugars, how you make fatty acids, all of the reactions in that process are catalyzed by enzymes. And what's amazing about them is that even though chemists, we understand a lot about how the catalysis occurs, but enzymes can do it much better than we can do it as chemists. So they accelerate the rate of the reactions by factors of up to 10 to the 16th. So that is a huge rate acceleration. So we, as enzymologists, look at organic chemists and say, eat your hearts out. Because they even understanding how the enzymes work, and they involve big protein structures, a chemist can't make something little to be able to recapitulate the rate accelerations. So I'm interested in enzymes that catalyze unusual chemical transformations that go on inside our body. And most of the enzymes are focused on making the building blocks required to make deoxyribonucleic acid, or DNA, the genetic information. And I became interested in those because not only because of the rate acceleration, but they have unusual chemical mechanisms by which they work.
Yeah, so tell me a little about that. I mean, why are they able to do this so quickly?
So normal catalyst, an organic catalyst or an inorganic catalyst would be a small molecule, molecular weight 500. Enzymes have molecular weights 100,000. Okay, so they're much, much bigger. So you have this big, huge protein, and where all the chemistry happens is in a little part called the active site. That's where the substrate gets converted to product. So why do you need such a big, huge protein? And that's because everything is coordinated in an amazing way. It's evolved with time. They start out, they're not very good, and over billions of years they become better and better and better. And what they do is they can align everything up in the active site where the chemistry happens. And chemists can't recapitulate that with a small molecule. It takes this big, huge structure to be able to do that. And so this alignment and the amino acid side chains in the active site, or metals, or organic cofactors, all can allow us to catalyze these rate accelerations by these huge amounts.
So was this what got you interested in this question?
I've had a weird history. I got interested probably not as an undergraduate. Biochemistry was not at the molecular level back in those days. But when I went to graduate school, I took a course that I couldn't get credit for because it was in the biochemistry department at Berkeley. And I had two faculty members, Koshland, who later turned out to be the editor of Science, and Kirsch, who was a physical organic chemist. And they sort of got me interested in enzymes. I really didn't know even what an enzyme was, and I was always interested in how they worked, how you converted this to this, what were the details of how that reaction happened. And I didn't know very much about proteins as catalysts at all. And by taking that course, I got really interested in this area, which is now sort of biochemistry.
Right. So so there's been sort of one, as I understand it, one enzyme in particular that's or probably several, but that have been really your focus.
So I worked on many projects, but the one that I won all or received notoriety from is an enzyme called ribonucleotide reductase, and they convert a nucleoside diphosphate. So the nucleoside diphosphates and triphosphates are the precursors to ribonucleic acid, or RNA, to a deoxynucleoside triphosphate, which provide the building blocks for DNA. So what's unusual about the enzyme is one enzyme makes 4 different building blocks, the ones you see in DNA, A, T, G, and C. And so controlling that enzyme, not only is the chemistry interesting of how the reaction happens, but controlling this reaction is really critical, because if you're making the 4 building blocks, if you make one 100 times more than the next one, then what happens is when you build them into the big polymer, you end up getting mutations. You put the wrong nucleotide, deoxynucleotide in, and then when the DNA gets replicated, you have a mutation. If you get enough mutations, you have serious problems. So this one enzyme that makes these 4 building blocks has been fascinating because all of chemistry was unprecedented, and we're still finding new things about it 35 years later, okay? But also the regulation is central to controlling nucleic acid metabolism, and that's why it has also been an excellent target for design of therapeutics. So, its central role in nucleic acid metabolism has made it easy to get funding. Not easy, but feel easy to get funding to work on this system for so many years.
So maybe you can sort of unpack that a little bit, for me. I mean, so the idea is if you can control how this happens, you can control undesirable or prevent undesirable mutations?
Well, you can I don't know whether they're desirable or undesirable. A lot of the mutations might be neutral, doesn't do anything. So, but if you have a lot of mutations, then one of them's bound to hit something that's important, that's critical. And what's interesting about this enzyme is it uses chemistry that involves free radicals. And free radicals you see in the press, you see it in the newspaper. I guess nobody reads newspapers, but you see it on whatever people look at now in the internet. Radicals are highly reactive and challenging to control, and so people eat the vitamin C or vitamin E, trying to cut down on the amount of free radicals, which are produced basically from our environment, like ionizing radiation or oxygen with reaction of metals that are found in our body can generate radicals. So I think at the time I started working on this, there were almost no proteins that involved free radical chemistry at all. But this enzyme that plays the central role in making the building blocks for DNA uses really complex free radical chemistry that was unprecedented in many aspects. And its nature has figured out how to harness the reactivity of radicals to really do extremely difficult chemistry with exquisite specificity. So one enzyme can turn over 1 million substrates into products without making a mistake, which is quite remarkable. So people are interested in it because we're controlling the relative ratios of deoxynucleotide pools, as well as this complex free radical chemistry, which could, if it goes astray, what happens is the enzyme kills itself.
So how does this translate into sort of drug therapies and things like that?
Well, you know, there's now a drug used clinically called gemcitabine, which sort of was, there were two drug companies, Marion Dow Merrill and Lilly, both working on this problem, and I was collaborating with Marion Dow Merrill, and they both made the same compound, and the one I was collaborating with lost the patent rights. But anyhow, it came out of, my collaboration with them came out of the fact that we wanted to understand how the substrate got converted into product. What is the detailed mechanism? And then one of the ways you study that is you have a structure and you remove one of the hydroxyl group or some small part of the molecule and replace it with something else. And that's what we did. And the thing we replaced it with turned out to be a mechanism-based inhibitor. That means that it looks just like the normal substrate, so it can bind in the region where the chemistry happens, but it doesn't do anything. It's only when the enzyme starts doing something on this small molecule. It does the first step the same as the normal substrate, but then it does something different and all hell breaks loose, and the enzyme ends up killing itself. So that's called a mechanism-based inhibitor, and gemcitabine is a mechanism-based inhibitor. The whole substrate blows apart. Now, the cytotoxicity mechanism is more complicated than just targeting ribonucleotide reductase. It kills that enzyme, but it also, as with most therapeutics, interferes with other things like DNA polymerase as well. So
it's effectively interrupting this process.
Okay. So what it does is, to make DNA, you need all your deoxynucleotides. So by inhibiting the enzyme that converts the RNA-like molecule into the DNA molecule, you then knock down the whole pool size of all these deoxynucleotides. Now, this drug, gemcitabine, looks like one of these deoxynucleoside triphosphates. So you've knocked down the normal pool size of deoxy-ATP, deoxy-GTP, whatever, and now you've taken this drug and made it into, it turns out it gets metabolized into a nucleoside triphosphate that then can compete with the deoxynucleotide pool, which has been drastically reduced, get incorporated into the growing chain of DNA, and it causes chain termination and replication. So it's a synergistic effect that you inhibit ribonucleotide reductase, and you also inhibit DNA polymerase.
So it effectively stops cancerous replication?
And it does it to normal cells too, right? So the question is, so when you deliver a drug like a nucleoside, if the substrate for the enzyme has two phosphates on them, which are charged. Okay, so to get things across our cell membranes, it's got to be non-charged. So you deliver the nucleoside, and so there are also enzymes that stick on 1, 2, or 3 phosphates inside the cell, and those enzymes have different regulation in normal cells from cancerous cells. So you have a therapeutic index, which allows this drug to more effectively target tumor cells than regular cells. But of course, it doesn't target all tumor cells because metabolism in each cell is different. And second of all, I've been in this area forever. I was even in a pharmacology department trying to rationally make drugs. Cancer is a tough problem, and it's very heterogeneous. And I think we shouldn't be spending so much money on making things like gemcitabine as a taxpayer. I think we should be trying to figure out how to diagnose cancer much earlier, given that we have all this genomic information, and then monitoring, figuring out what the signs are that you may have a predisposition to this, and then dealing with it at a much earlier stage. I think dealing with cancer at the very late end stage, where gemcitabine is used, it's used for treatment of pancreatic cancer. Frankly, I'm not sure I would take this drug, but it makes billions of dollars. So
in the case of something like pancreatic cancer, if you were able to detect it much earlier, is there something that you could do to...
Well, what you do is cut it out. Okay, or hit it and kill the cancer early. What happens is most of the time it's already spread everywhere, and by then it's just too late. And so I think at the end, if you know anybody at the end stages of cancer, most of people they really are in a lot of people are in a lot of pain. So I mean, what you want to do is diagnose early. So I don't know. Cancer, if I, as a taxpayer, I would be putting my money into Alzheimer's disease, not cancer. That's just I mean, if you look at everybody has to die of something when you get older, unless they find some magic bullet, which they probably will 50 years from now. But, if you look at Alzheimer's disease and how long that lasts, if they could figure out how to prolong the onset of that, then you would hopefully end up dying of something else that wasn't as horrific as Alzheimer's disease. Yeah.
Right. Interesting. Yeah.
So anyhow, I mean, I think to me, ribonucleotide reductase, that there's a small company that has just made the first antibacterial agents, and I think that's a much better target and something where you can clearly kill the bacteria and not touch any human system. And we discovered a couple of years ago that there's unusual metal cofactors in almost all the pathogenic bacteria. And it turns out, this company has found the first inhibitor. It's now just starting clinical trials. They're getting ready to publish their first data. And I actually think you could rationally design inhibitors of pathogenic organisms. And given the issues we have with resistance, which are quite substantial, I think their ribonucleotide reductase would be a great target for Staph aureus or M. tuberculosis or whatever, a number of the other organisms. And you don't have to worry about all these cytotoxic effects. And furthermore, you don't have to use nucleosides. So this drug gemcitabine is a nucleoside. And as I sort of have already explained, it's complicated because it's got to be modified inside the cell to act. It has multiple targets inside the cell, which most drugs actually do have. But now with bacteria, there's a specific target unique to the bacteria that we don't have in the humans. The metal center in the bacterial systems that's required to do all this radical chemistry is distinct from the metal center in the human. And so, and there are biosynthetic pathways by which these metal centers are made, and I think you could inhibit those centers. So that's one of the things I plan to do when I retire. That's one of the things is just go back and work in a lab and put together a group of people in this area that specialize in a particular organism, like tuberculosis or something like that, and see if we couldn't make specific inhibitors of ribonucleotide reductases. Yeah.
No, it's fascinating that it's taken these two different routes.
Right. Well, I've always been interested in antibacterial things forever. I mean, to me, people went away in this country. People went away from studying bacteria for years. It's now come back because of all the resistance problems. But it's amazing how little we know. We know we've got the whole human genome. We now have thousands of bacterial genomes. But it's amazing how little we know about the biochemistry of cell wall biosynthesis. It was studied in detail in the 1960s by, the first paper on how penicillin worked was by Tipper and Strominger in 1964. That's one of the things that got me excited about biochemistry, actually. And now there's two groups at Harvard, one in the chemistry and one in microbiology, that are really doing beautiful studies using all the amazing technology we have and setting up screens so that we can target cell wall, which is unique to the bacteria, which has been targeted forever. There are a lot of natural products, but now we can actually target other aspects of unique metabolism in bacteria, which is really sort of what you want to do, and ribonucleotide reductase would be one of those targets. So,
wow. That's fascinating.
Yeah.
And tell me about the sort of the plastics side of things.
Okay.
How does that come about?
So that came about because when I first got here, I met Tony Sinskey, and I met him because he was interested in catalytic antibodies. So I used to, not that I was interested in catalytic antibodies, but they catalyze reactions, so I could help him think about stuff like that. It turns out he was one of the people that first discovered an enzyme called polyhydroxyalkanoate synthetases, which take a small little building block, 3-hydroxybutyric acid, and make them into huge polymers. So synthase just means they put many of them together. But what's amazing about them, man can make these polymers as well, but bacteria can make them much bigger with uniformity. So when and nowadays man is getting better and better at making things uniformly. But when I started working on it, they couldn't do that, and so at the time they were interesting because if you mix them, two of these building blocks together, you get the polymers had properties of thermoplastics, yet all bacteria have enzymes that can degrade these things. So the idea was, could you make them economically competitive with oil-based polymers like polypropylene or polyethylene? And I was interested in the mechanism of how they worked. How do you do initiation, elongation, termination, and how do you control all of that? And it turns out, like, if you grow the organism that Tony worked on, and we were involved in the first paper that purified the protein. The proteins were really hard to purify back in those days. What they do is, if you feed the bacteria the right thing, 85% of the dry cell weight of the bacteria can be these polyoxoesters, and they form these big granules inside the bacteria. So even under a light microscope, you can see these polyoxoesters. And I became interested in how that process worked from a biological perspective and from a chemical perspective. And actually, in collaboration with the Drennan Lab, we just had the first, we've been after this for years, we just got the first structure, it's not even published yet, of this synthase. So the mechanism of how it works, non-template dependent polymerizations as opposed to DNA and RNA biosynthesis, which are template dependent, is still to me a major unsolved problem. And it's not unique to polyoxoesters, but sugar polymers like in the cell walls, actually, again, of all different kinds of bacteria, tuberculosis, but all have these polymers of sugars, and the initiation, elongation, termination processes are not really very well understood. So I became interested in that problem, and am still interested in that problem. So,
yeah. So and it presumably holds all kinds of implications.
Okay.
But I mean, if you can replace petrochemicals with
yeah, so
right?
if you could do that. So Tony is he's a typical MIT person. So he's a bioengineering plugged in. He works on all kinds of things like whatever the biofuels that he's working on now. Yeah, so the idea was he started a company focused on this. And I think to make the issue, I think, and again, I'm not an engineer, so I don't know how to translate this into something that's economically competitive, but the issue is you want to get these biopolymers out. You can make a lot of it without using extraction with organic solvents or whatever you do. And so the company really was focused on trying to make these things in plants. And they made, I don't know how much, I don't think they made very much money, but they made things like there was sutures or stuff that are biodegradable. They had biodegradable shampoo bottles. Monsanto put a big effort into this, but in the end, I think they were missing. Again, my idea was not to make something commercially viable. I was just interested in the basic mechanisms of how these things work. But to me, what was missing in this whole problem was how you control polymer formation inside the cell. And there are proteins, other proteins besides the polymerase. And I think if you understood that better, you could do it much better. And so that's what I was focused on, trying to understand how the coordination of not only the polymerase, but also the other proteins that were identified. Now they're identified because we have genomes of everything, and they're usually organized in what's called operons, and that means all the genes involved in a biosynthetic pathway are contiguous in sequence space. And so yeah, we spent a lot of time trying to understand all that stuff from a more biological point of view. And Tony is a microbiologist, and so I learned a lot of microbiology from working on that problem that's now routinely used in my lab. Yeah.
So yeah, I'm interested because you mentioned you're sort of not interested personally in the commercialization.
I know this is a terrible thing to say at MIT. Everybody looks at me like, whoa, where's this woman coming from? What's driven me all the time is just like trying to understand how the enzymes work. That is what, and if something everything I've worked on has been practical. From that, I worked on drugs, natural products that cleave DNA that are used clinically. So everything I've worked on, I sort of know that you need to do that, but really what I'm interested in is how they work. And then if you understand how they work, can you take advantage of that understanding to make something better, or to rationally describe how you would make this better, and then find the right collaborator. So if you have to synthesize something and it's hard, I can make things, but not things that are this big, then you find a collaborator and convince them to do things. And so I would say, my whole career has been spent with smart collaborators. And without those smart collaborators, I couldn't have done anything.
Yeah.
So every place I've been, I've had really good collaborators. And almost all of them have worked. And that means you just have to find the right person that you can talk across disciplines and you have fun. And there's no ego involved. You just have fun doing the science. Yeah.
Yeah. So how? If we could go back, and talk just a little bit about, where your childhood or wherever.
Yeah.
Were you interested in science as a kid?
Not this kind of science. Actually, biochemistry didn't really exist then. I was always interested in nature outside, so we had a shack on a lake in New Hampshire, a tar paper shack. It had bats underneath it. I used to take a butterfly net to bed at night because the bats used to swoop over my head, and I would catch them and let them go. Anyhow, we spent a lot of time outside on a lake and in the woods. I knew where all the flowers were. I knew where all the birds were. I knew all of that stuff. I used to fly fish with my grandfather. So I've always loved the outside and sort of the nature part of it. I still like that. One of my hobbies is gardening, but gardening with just flowers, that kind of stuff. So I guess I was always interested in nature, and rocks and whatever. I had a nature club when I was in the 6th grade or something. Not only the 6th grade, through several grades, I had a nature club and used to be the teacher that made them do stuff. And they'd have a prize back in those days for who could find the most wildflowers and name them. We always won. Anyhow, so that's where I think I got interested in all of this. And my first introduction to chemistry was in high school. And I had a really good high school teacher, Mr. McHugh. And what I liked about chemistry was the colors and the crystals. That's what I liked. So I was excited about chemistry, and I was lucky that my father was a faculty member, and he got me a job in the lab. I had some other kinds of jobs in factories too, but I had a great job in a lab, and that's where the person that I worked for, Ed Trachtenberg, who was at Clark University, taught me organic chemistry before I ever went to college. And I just loved messing around in the lab. So I like details. I like to put things together. I like to see the changes. I really love working in the lab. So that's where I got excited about it. And when I was an undergraduate, I took chemistry. I was interested in math. I was interested in art. I was interested in art history, interested in a lot of things. But again, I had a really good mentor who was also the guy at Clark University. Trachtenberg was a physical organic chemist, and the person at the University of Pennsylvania was also a physical organic chemist. And I would say they had a big effect on the direction that I went in subsequent to graduating from Penn. Yeah.
And you say your father was a faculty member. Was that was at Clark?
He was at Clark and also Worcester Polytech. He was a mathematician. So, I mean, it was like, I was a nerd from day one. I mean, that hasn't changed. I mean, I used to be the one, everybody else is doing all this stuff, and I'm sitting in the corner reading something. I mean, that's just the way I've always been. Antisocial. Whatever. Just immersed in what I found exciting. And it's still, I mean, what's amazing is I've been able to do that my whole life, right? Just go home. When something is depressing, or, everybody has depression once in a while, I just go read somebody's paper that I think is exciting, and I can feel the adrenaline pump up through me. Even now, my lab is winding down. It drives me crazy because it's just not enough results. I mean, I love results, right, to think about them. But, I go read one of my friends' papers. I have really smart friends, and I just get excited, or they have me come over to group meeting and we just have a scientific discussion. So that part has never gone away. I just immerse myself into something that I find interesting and can get just as excited about that as my own stuff.
Sounds great.
So, yeah.
So you didn't plan to go into chemistry then?
Not
or
really.
something, really.
No. I don't even know. I mean, I guess I sort of liked chemistry a lot. And I worked every summer. They had a National Science Foundation fellowships I worked in the summer. I didn't know whether I thought I wanted to be a teacher. I tried teaching in high school in Worcester. I grew up, and, this was like after I was substitute teaching. And I had 2 kids pull knives on me, and I had to call the principal. And I couldn't control the class at all. I mean, I had no education courses, and so I decided, well, how can I teach? I tried being nice. I tried being terrible. I couldn't talk to the students, and so that's when I decided, well, maybe I need to go do something else. So I couldn't decide where to go to graduate school, and so I just they didn't have an internet. They really didn't have, they had no faculty advisors. I mean, that stuff didn't exist back in those days. But I was talking to Thornton, who I did undergraduate research with at Penn, and he said, you need to apply to at least one good school. And so, I had chosen schools that I thought were good, but of course I knew nothing about the chemistry departments. And so he got me to apply to Berkeley, and that's where I ended up going. And if I hadn't gone to Berkeley, I wouldn't be here. Yeah.
How so?
So, because it's an old boy network. And if you don't go to school A, B, first of all, if you're a woman back in those days, forget it. But if you didn't go to this school, this school, this school, this school, it's really hard to make it into a place like this.
And is that still the case?
I think that's still the case. I still see name prejudice big time. Yeah. It's getting better, but I think it's still, and it is there for a reason, but the reasons really are not so compelling anymore. So when I think back on it, I mean, I certainly didn't think about anything like that. I still thought I wanted to teach because I think that, you sort of affected by, my mother was also a teacher in high school, so you think about this, you're affected by teaching, and that's what I thought I wanted to do. I tried it in high school. I was completely unsuccessful at it. And so I thought maybe I could teach in a small liberal arts school somewhere, and that was one of the things that I started. That was my first job. My first real job was in a small liberal arts school, yeah.
After Berkeley?
After Berkeley. Well, I did a postdoc, and while I was doing a postdoc, I met a guy who was on sabbatical from Williams College, and they had a job opening, not for someone trained like me, but he got me a job interview, and they liked me, and so I got the job, and that was my first job.
And what were you teaching there?
I was teaching, well, I was in a chemistry department, but I was teaching physical chemistry and biochemistry, neither of which were my specialty. That was baptism by fire. That was really, Williams was an interesting place, really smart undergraduates, but it had been all male since the late 1700s. I was the first female anything in science. It was the first year where they had just gone co-ed. So that, in addition to teaching stuff that really was sort of outside what what I knew at that stage, which probably wasn't very much, I mean, was tough. But I think what it did was gave me enough self-confidence to take the next step. Because, when I started there, I didn't know anything, but within a year or two, I realized how much I liked science and how much I wanted to do science, and that I could use my energy to get undergraduates. You could tell them anything, and they got excited about it back in those days. And so, I worked with a lot of undergraduates. They would be in the lab at 2 or 3 in the morning with me. I mean, it was really fun. They all published papers. I had an NIH grant, all of that. And that gave me enough self-confidence to take another step, I think.
So that was at Williams
That
College.
was at Williams, yeah.
And so what was the next step then?
Well, to get out of a place like Williams, okay, there's no way you go into a big research place going out of an undergraduate school where, there aren't any PhDs, or I think they had a master's program, but there was nobody that had master's students when I was there. I did another postdoc, basically. And so that's when I went to Brandeis. And then at Brandeis, I get more self-confidence, and so I applied. And that's where I really became excited about enzymology. I work for one of the smartest people in the world, I think, one of the most creative people in the world. So, I mean, I just couldn't, I just loved it. And I was surrounded by really smart postdocs. And so for me, it was just, I had a wonderful time. I had taken a leave of absence from Williams and decided not to go back. So then I applied for jobs, and I got a job in the pharmacology department at Yale because I thought I was interested in rational drug design. So understanding something in detail, taking advantage of that understanding to create something that would kill an enzyme, a mechanism-based inhibitor. That's what I was excited about. And Yale was at the forefront of that. It was a really good job. But I think rational drug design was maybe 5 years too early. That's what I think. So there were no there were 50 faculty and 4 graduate students or something like that. So for me, it was great because I love working in the lab. So I worked in the lab all the time, and that's where I made the key discovery. I mean, I can still feel the adrenaline from that discovery
Well, tell me about that. How did that happen, and what was that like?
Well, I mean, it was just, it's like I don't know if you've ever read the book Lucy, where you have an archaeologist finding bones in the desert in Africa. I mean, if you read his story about that, I can't remember the name of the guy, it was the same thing. So you're working on this thing, you don't understand how it really works. And then you've done an experiment, something is going on interesting, and then you do an experiment, and then I put it in the instrument, an NMR instrument, and I saw something, and immediately all the lights went on. I understood how this basic reaction that nobody understood worked. So that one signal I saw coming out of this instrument sort of gave me a really good working hypothesis. You never really know how they work, but we had a really good model for how it worked. And that really was a discovery that allowed me to move from Yale into another place where I could have graduate students.
Yeah, so it's interesting. So really, because, well, it really was a moment, it sounds like.
I've had many moments like that. I think if you work, that's what's different. I mean, most people have big labs. They don't really look at things on a day-to-day level. And they have here everybody has this big picture. I sort of have a big picture too, but I like the details. And I think it takes both to make science work. And to me, it's the details that they don't see. I mean, working in the lab, do you see something? And it's these things you see, and then you chase. Do you have the intuition to chase it? That really sort of make all the difference, I think. And so, in my labs, the kids really have ad nauseum detailed group meetings. Because I really, and they can show you data, it looks beautiful. But when you ask a few questions, you quickly find out that, nowadays people can plot anything anyway. I mean, and it looks nice. The question is, what is this plot based on? And so, yeah, anyhow, I like the details, and in my lab, that's been key to my discoveries are the details.
So you say that sort of propelled you onwards from Yale.
Yeah,
Where,
so what happened at Yale, I mean, that discovery really put me on the map, for whatever map it was, but I think it was, again, a self-confidence thing. So I started at Williams, I had no self-confidence. I took the next step, and I made this discovery, and somebody thought it was interesting, and that got me the job at Wisconsin, which I think was in the biochemistry department. So I've been in chemistry, pharmacology, biochemistry. So I really have sort of been on this interface my whole career. And back in those days, it was tough being on the interface. Now everybody's on the interface, but it wasn't. Anyhow, I was grateful to have gotten the job at Wisconsin because there were they made so many seminal discoveries about how vitamins worked in the biochemistry department. And so again, I mean, the teaching, biochemistry, mostly you were a chemistry major with a course your senior year in biochemistry. So you really didn't have that much teaching compared to chemistry departments where you have a lot of teaching. So I got to work in the lab all the time. So, and I did. And, all the projects that I have succeeded in chasing down over the years. There are 4 different areas I've worked in. I've worked on all these projects. And I think that makes a huge difference because you have some intuition about what can work and what can't work. And so when somebody tells you something isn't working, sometimes you have intuition that it's going to work if you just try a little harder or something like that. Yeah. So, I mean, that was the first place I got graduate students. So what that allows you to do is work on more than one thing or have a chance to have a bigger impact. If you're working on something that's really competitive and you have 2 or 3 people, it's really hard. But my lab has always been small, and that's because I like to pay attention to what's going on, and I'm not capable of paying attention to what's going on with 15 or 20 people. I just can't keep it all in my brain. So
yeah. So you say it was hard to sort of be on that interface of biology. Why was that?
Because I think chemists thought all biologists were morons. I couldn't get, when I was at Berkeley, there was no way I could get credit for taking this course with Koshland and Kirsch, who were trained by the best physical organic chemists in the world. Okay, because they just thought, and it's true if you look back in those days, okay, because it wasn't quite at the molecular level yet. You would take a course in basic biochemistry, and they would draw a structure of glucose or something, and it didn't look anything like glucose. Okay, so that's where they were coming from, and then biologists thought you didn't know, here I am on the interface. I thought I didn't know any biology, which in part is true too. Biology is really complicated. So being on this interface, that's why you need collaborators. So, I never did genetics, but starting with Tony, and now I've collaborated with somebody in yeast genetics, we do that simple genetics in my lab. So it's this cross-disciplinary thing, which everybody does now. You can't survive without doing it. But I think back in those days, it was much more challenging. Physical organic chemists, which is what I was, were studying the details of how things work, but they didn't make the move into enzymes. And so the field sort of died. Okay, I'm now at the stage, enzymology is at that stage. So my students, they wouldn't call themselves enzymologists at all, because everybody thinks we understand everything about enzymes, which is clearly not true. But now what are they doing? They're using all these basic principles to do engineering inside cells. Okay, so they've made another jump. So you have to keep, over the course of my career, I've reinvented myself many times, and probably the most important reinvention was at Wisconsin when, this is in the early 80s, when molecular biology and DNA sequencing, where you used to use these big DNA sequencing gels. I mean, they were a nightmare anyhow. And I learned molecular biology just by going to seminars and realizing what you could do with it. And so then I started reading about it and then incorporated it into my own work. So you have to keep moving with technology changes. And I think that wasn't so hard, in my career. But I think it's getting harder and harder and harder because the changes are coming faster and faster, and there's nothing built in to allow you time to relearn something. And some of these things are extremely complicated, and you need to look at the details or you don't use it right. So what I see now happening is people using all this technology, which is amazing, except they have no idea what's going on. That bothers me a lot, because that means you're probably not using it the right way. So I don't know. I think this is something where we need a reset button somewhere. And for me, over the course of my career, I did take sabbaticals. And I took sabbaticals. I did a sabbatical where I did one in X-ray crystallography, one in yeast genetics. I did one in Lippard's lab in inorganic chemistry because all the metal clusters and ribonucleotide reductases were there, and I needed to learn about metals. So I was able to take advantage of that time off to actually get into the laboratory and learn how to do a lot of these methods that I knew that I wanted to bring into my own lab. Nowadays, I just think it's harder and harder and harder. Mm-hmm. So I don't know what people are going to do, but it's different.
Yeah. Now, as I understand it, at Wisconsin, you were the first woman tenured in that department. Is that correct?
In biochemistry department.
Yeah, what was that like?
I don't know. I didn't even think. I never. Do you know that this is like amazing. When I came up for tenure, first of all, I was an assistant professor for 12 years. Okay, I didn't even know it. I didn't even pay any attention to it. When I came up for tenure, I didn't even know they were voting on me for tenure. Somebody came in and said, "You have tenure." I mean, that's because I didn't feel, nowadays these kids, they come in, they're completely, from day one, are focused on tenure. What do I do? Can I get my paper published in Science and Nature? I mean, I didn't go through any of that. So I was completely oblivious to the whole thing. I mean, I had a really nice lab. I was surrounded by really good colleagues. Wisconsin in biochemistry, extremely strong in enzymology. I taught with one of the smartest kineticists in the world. And learned a tremendous amount from my teaching with him. So I love Wisconsin, But I didn't know anything about tenure. I didn't think about the fact that I was a woman. I did think about it when I was in graduate school, but I did not think about it subsequent to then.
Yeah.
And
so then how did you come to MIT?
So the only reason I came to MIT was... This is really not. Probably I shouldn't be saying this, but anyhow, the reason I came to MIT is my parents and my brothers and sisters lived in this area, and my parents were getting older, and my siblings perceived that I didn't have a real job. As a teacher, you didn't have to do stuff most of the time. You taught a class, and then you could go do anything you wanted to do. So they thought it was a good idea if I came and helped take care of my parents, who were aging at that stage. And so that is the reason I came here. And I had done a sabbatical in Lippard's lab just because there were two people that were the best in the world in that, and his lab was one of them. And that's where I decided to go. And I think they liked me when I was here. And so I think they had horrible luck with women in the chemistry department here. And so I'm sure I was on people's radar screen because they needed to hire a woman. But that being said, that's chased me my whole career. It doesn't matter what you do, right? If something good happens to you, it's because you're a woman. Anyhow, I got the job, I think, because I was on sabbatical and I interacted with a lot of people, and people knew that I really liked science, and they thought I hope they thought what I was doing was interesting. And so that was in 1984. And so my parents, plus the fact that this was such a good place, is the reason I chose to come here. But Wisconsin was a fantastic place to do science. It was a kinder and gentler place than MIT.
Yeah, it's interesting you say that being a woman has chased you your whole career.
Yeah. Well, it is. I mean, for example, this is something that should get out there. But when I would win an award, I never told anybody Why? Because the immediate response from your quote-unquote friends was that you won it because you were a female. So, I mean, that's just something, it just sort of took whatever the thrill out of something good. It was, people were jealous. I mean, I think that's true with males too, but I think it was even worse with females. So anyhow.
No, that's terrible.
I mean, to me, I'm just grateful. The awards don't really mean that. A few of them meant a lot to me, but really, to me, the real thrill was just being able to do my own science. Yeah, get paid to do it.
Do you think things have changed at all with regard to sort of the gender issues here?
Yeah, I think they're much better. I still think they exist, but I think they're much better. I mean, since I've been here, you have a lot of. I mean, if you were a female chemist and were pregnant back in those days, forget it. That was the kiss of death. There was no way you were serious. I mean, I think the whole country needs to move another step forward. They need to take a look at what's going on in Europe, because how do you deal with all of these issues? I think MIT has done an outstanding job at that because of the Hopkins committee, which I was on in the very beginning. Yeah.
Oh, is that right? You were on.
I was on the original committee. Yeah.
Yeah, that was extraordinary.
Yeah, I mean, well, I think I was treated fairly, but they clearly, I mean, we had data that showed things weren't correct, and I think it did. It put it in the face, and both Bergeneau and Vest were way ahead of the time, and still I get asked to be on a lot of these reviews, and it's not moving anywhere near that fast in a lot of other places.
Right.
So.
Right. Yeah. So how is MIT different in some ways that have helped you in your career? Say, different than places like Madison.
I think the name. Yeah. I mean, I think just the name, that's enough. In other words, if you have the brand, gives you visibility that you don't have with a Wisconsin, for example. I mean, it just puts you right up there, out in front. I mean, to me, what's been the great thing is the collaborations that I've had here, so that my science has also been much better.
Yeah. So, so tell me about teaching. I mean, we haven't talked much about that. I mean, because you talked about your early experience with teaching, but something changed.
Right.
So.
Well, no, I thought I always wanted to be a teacher. It was just that in high school, I couldn't handle the discipline problems. So, of course, in college, kids just don't come to class. So you don't ever have that problem. So I think teaching, teaching is right now undergoing a revolution. So what I think or thought about teaching is probably not all that important, but since I've been here in chemistry, there was no course in biological chemistry. So I and Bill Worm Johnson and John Essigman started the first biochemistry course in chemistry. There was a lot of kickback from that in the biology department. That was not so easy, but now I think probably this fall we're going to have the first course, 5, 7, major. So I think it finally got there after all of these years, that we've come to some kind of common consensus, which we should have had a long time ago, in my opinion, where you have people from biology teaching with people from chemistry. They have different perspectives. I just think it would make it much more exciting. But, those are the lecture days. So I don't know what's going to happen, because the lecture days, are they numbered? I don't know what's happening with all of the online stuff. But anyhow, we sort of invented 507, and I would say if you compared 507 with 705, which is the biology counterpart, it was much more chemical. That's what I brought to the table. How do the vitamins on your vitamin and your vitamin bottle work? How do enzymes work? So it was much more molecular and detailed, although we still had a lot of metabolism in there. I invented a second semester of this, which was, this is 20 years ago now, on macromolecular machines. And I think, within a decade, everybody was doing that. So I think that was way before its time. I taught a course with a physical chemist, organic chemist, and me, and I would make up. This was a capstone course where I would choose a target, like hepatitis C or tuberculosis. And as a team of people, so these were, 20 people, we would have different groups, and I picked the target, like mechanism-based inhibitor of this target, but we chose a lot of different targets and different approaches, and then they would work in teams and talk to each other in 5-minute bullet points about what they were doing, and we would come up and design a specific inhibitor of what the target was, and that at the end of that, they had to defend it against people from drug companies in this area. And the kids loved that, but they had to work pretty hard to do that. So I would say that course and my enzymology course, which I think was outstanding, are sort of what I focused my energy on. And then I like the lab. I invented the first biochemistry lab. We had this is many years ago now. Even though I wasn't really teaching that, I helped put together that lab so that students could learn how to isolate proteins and assay proteins. So teaching I love. I actually like being in the lab, walking around and talking to the students to make sure they really get what they're doing. They're not just following some recipe. I like interacting with undergraduates at MIT because they're really smart and they're always pushing you. And so that for me has been really exciting. And they come back to you. The really smart ones come back to you, your office, and they bring you a paper and they want to talk about their research. I mean, all of that I think has been a thrilling part for me for being in a place full of such smart people. So I think teaching is extremely important. And one of the things I thought about when I was going to retire was, should I go back to a place like Williams? But I don't know what teaching is going to be now.
What do you think? I mean, what you mean with the digital learning and all that?
I don't know. I really don't know. I mean, so I can tell you the best. We have things for our graduate students called tutorials, and when new graduate students come in, we have problems in kinetics and thermodynamics and metabolism, and we limit it to 5 people and a faculty member, and that's where they learn the best. So they have problems. They all go to the board. In general, once you break down the barriers or the nervousness, that's where the kids learn the best. But how can you afford to do that in terms of time and faculty members? So I would say for me, that's been the best learning experience. And what you see is the students help each other, and you're there sort of to keep steering them in the right direction. And that's what you would love to have in all undergraduate, but I mean, it's impossible, I would say. Although they do it at Oxford, right? I mean, Oxford and Cambridge has done this forever, that you have a tutor or something that you learn from. So I don't know what's going to happen with the digital thing. I mean, to me, it's, you walk up and down. They don't do it in my class, but if you look at a big class, I mean, all the kids sitting in the back are playing on their computer. A lot of kids don't come to class anymore. So, I mean, when I first started teaching biochemistry, 95% of the kids would come to class. And I think I stopped teaching with Essigman in 2013. Only, I would say, at most 60% of the kids would come to class. And Essigman is he was the best teacher in our department. So, I mean, they don't learn that way. So that's the issue, is how do students learn? And they don't learn the way I learned. So once you lose that connection, how do you teach them?
Right.
So I don't know. I mean, I've been going through up and down about that. How is it that you connect? I even tried playing Lady Gaga songs. They got stuck in a bad project. Nothing. I just feel like I couldn't grab their attention. I mean, in a class of 120, you always grab 20, right? I mean, because they're passionate. It wouldn't matter what you did. They would be passionate about biochemistry. How can you not be passionate about biochemistry? It's everything that's going on inside of you. But it's the other 100 that how do you connect to them? They should all be excited about this.
Yeah, that's a challenge.
Yeah, it's a challenge. For me, it's a challenge, and I don't know what, to me, that was one of the things I thought, well, I could go back and do this because I really love biochemistry. And I mean, so if nothing else, they can feel the energy of how important this is and how exciting it is. But I just think it's changed. So
Yeah, yeah. Well, I think the enthusiasm and all that still,
yeah,
still must be vital.
the enthusiasm I think is vital. But I would I would think most people teaching here that cared about teaching are really, you see that enthusiasm, passion. Yeah.
Now tell me about, you mentioned art history and
Yeah,
and also I
yeah.
read you taught an archaeology course or co-taught.
I did.
How does that come about?
So that came across because when I was an undergraduate, I was really interested in art history. So I would take a course and somebody would start talking about something, and I'd go to the library for 3 weeks, read about some artist I was interested in. And so I was and I can just remember the first times I saw real paintings, and I became fascinated as to how you cleaned the paintings and how you authenticated paintings. And so one of the things I actually considered when I went to graduate school was to work for a photochemist. But that didn't work out for reasons I won't go into. But that's something that's always stayed with me, and I still read things. Like I remember when they were trying to clean the Sistine Chapel, and so you could remove the soot from the oil lamps on top of this, but how do you do it and then restore it to what it really looked like? Okay. And so that would become a real issue, because then you really had to be an art historian sort of to really understand what it really looked like. And I can just remember when I went to Italy once, they had a chapel in Florence where the altar was up against the wall on a fresco, and then so if you move that back, all of a sudden you could see what it really looked like. And so that served then as the mainstay for how you would go about what kind of chemicals were you going to use to clean all of this art, to restore it, and how do you authenticate things. That's where I became interested, how to use carbon dating and stuff like that too. So I've always been interested in that. And when I first got here, I taught a course over IAP on that. And, the Fine Arts Museum here had a pretty good restoration. And so we would go there, and they would oftentimes get a lot of the equipment from chemistry that we were discarding. And they could use like a mass spectrometer and stuff. They would use it to help them authenticate things. And the National Gallery, and then I'm drawing a blank, but anyhow, there's another place in England. Both of those places had really good books on all of these issues. So you could teach kids chemistry at the same time as keeping their interest in art, which some of them had. So and the other course I taught is chemistry, and I taught chemistry and winemaking. And that also was extremely popular. They had to learn a lot of chemistry. But they also got to make wine. So both of those I had a good time teaching. Yeah. And they were all sort of non-chemist types. Yeah.
And what about the archaeology course?
So the archaeology thing was to the dating part of all of this. Yeah.
Right.
So they had a few lectures on how you date things. Yeah.
Yeah. Oh, neat. That's neat. Yeah. So another thing, that I was interested in, I read that you're part of Friends of the American Junior Academies of Science.
I don't think so.
Oh, all right. That's misinformation then.
I don't even know what that is. What is it? What is that?
I was going to ask you.
Yeah, I don't know. I mean, so I would say in terms of being a mentor to people, I would say that I've been a mentor to people in chemistry over these years. I mean, if you look at the history of the biological chemistry division within chemistry, it's been tough. And it's now in a place where it's great.
It's been tough in what regard, you mean?
Because the way it was formed was, that division was formed because Deutsch did away with, I'm trying to remember what the name of the department is. They call it fruit and nuts. What is it? Does anybody? Anyhow, it was this nutritional whatever.
Food science?
Food science. I don't know, remember what the name of it is, but that the correct name should be in there. Anyhow, so there were a whole bunch of people in that, and he just disbanded the department when I first got here. And those people then either left or they were put into other departments. So in contrast with the other divisions within my department where you hire, and that's what we do now, of course, we automatically had this nucleus of people. We had a tiny little nucleus before, and then it got much bigger. And some of the people were, they were all really smart, but they chose not to be in a department like a chemistry department. So I would say it took us a long time to get to the stage where we were starting to attract, number one, good graduate students and also really outstanding faculty. And I would say that I've spent a lot of time mentoring people over the time period. And I think we're now at the stage where we have a fantastic group of people. So, yeah.
Yeah. And so what's next? You mentioned retirement.
Yeah.
So, sorry.
Yeah.
Yeah.
So retirement, I did a lot of thinking about this, and if you looked at the Welsh Award, you saw Chris Walsh, and he was a chair of the department in chemistry that hired me, and then he left immediately for Harvard Medical School within a month. Okay. Anyhow, we've been friends forever, and I was on all his kids' thesis committees and stuff like that. We used to talk about this all the time. So science is changing 100,000 miles an hour, right? I think most people, when they get up to be as old as I am, you still might be smart, but I don't think you're anywhere near it. I'm not anywhere near as good and can't remember stuff, and there's 10 times more to remember. I can't use it. I don't do science the same way as young people do it. They have a completely different style. I think this happens to every generation, and I think you need to know when it's time to let these creative people who are doing all these things you think is nuts, but that's what happens to every young generation. That's where all the creative ideas come from, in my opinion. Most of the creative ideas. So I think at the end of your career, most people around here do this with such intensity that to just cut it off, it's sort of like suicide, I think. But I think you need to know when it's time to give up your space for the next generation. And the question then is, how can you continue to do what you're doing, leaving the slot and the space open for the next generation? So I did a lot of thinking about this, and so did Walsh. I mean, he was 2 years older than me, and he was the top in the country. He gave up his lab 2 years ago. And I had decided this again so that I would finish in June. And, I'm dying for my science fix, right? I mean, I love data. But, I can get it from, as I was saying, I can get it from reading other people's papers or working and thinking with students about other people's systems, because basically I think almost everything I read and think about that's sort of biochemically oriented with proteins or enzymes or receptors, I think is exciting. So the question is, how can I do that once I retire? And that's what I'm trying to figure out right now. So.
And you mentioned possibly working in labs or.
Yeah. So what I think I'm going to do now is I'm going to try to organize some kind of a program project that might be funded by somebody like Gates or whatever, the Gates Foundation, and work on something like a disease like tuberculosis. So I was telling you I thought RNR is an interesting, ribonucleotide reductase is an interesting target. So, I put together Peter Schultz, who's now the president of Scripps, but anyhow, he's had all this screening stuff set up. I've known him since he was at MIT when I was here in 1984. There are people at Harvard Medical School that are interested in antibacterial systems. I want to do the enzymology, and then we need to get some tuberculosis experts, and we're in the process of trying to put that together. And I think maybe that's what Staph aureus is the other target. I think that's what I probably will end up doing, working back in the lab. The question is, can I do it? And I mean, up until, before I moved to MIT, I worked in the lab all the time. So up until age 40, whatever, I was working in the lab big time. And when I came here, the teaching load became pretty heavy. And so then and then the amount of money you had to raise also increased, so you had to spend much more time. And then your visibility, once you get in a place like this, all of that removes your time from working in the lab. And so I would say that's the reason I became a scientist was because I love working in the lab. But whether I can still do it, I don't know.
What do you mean whether you can still do
Well,
it?
it's physically exhausting. Okay. Also, everything you use in the lab now, while you understand how it works, there's a barrier to everything. So I don't know. It'll be interesting.
Well, you'll give it a try.
Yeah,
it sounds
give
like.
it a try. Right.
Are there other things you're thinking of doing? I mean, if you find yourself with more time.
Yeah, I know. I'd probably read something other than science. So I own a house in Maine that's on 3 acres of land, and it's got, it was built in 1750, and it has beautiful yard with all these wildflowers in it. So I don't know. I'd probably immerse myself in the history of the place where I am and get involved in some way with the gardening and just reading history, biographies, anything except science. What I found over the years is that I became so fixated on science that even if I read it 15 hours a day, I can't even come close to reading what I need to read.
Wow. Wow.
So I think it would be nice to be able to do something, something else.
Yeah, no doubt. Yeah. There's so much that we could talk about, but I feel like I've covered a lot of things that I know I wanted to mention. Are there things that come to your mind that?
Yeah. Well, I mean, I just would say that I'm grateful for having been supported to do my research by taxpayers for my whole life. And I hope, I don't know what's happening now. I'm worried about a number of things in the current environment. The funding, I think, is challenging. People spend a lot of time doing that. The young people spend way too much time chasing, quote, visible journals, on the other hand, that I think are terrible because they publish everything in size 9 font and you can't read it, and all of the science isn't supplementary information and you can't think about it. I think there's a huge amount of information out there, and I think how you use it and how you organize it in a way that's useful, I think, is a real challenge that needs to be discussed. I also think there's a lot less scrutiny and detail of scientific data that I worry about a lot. I don't think people look carefully enough at the data. I think people publish way too much. So I have very strong opinions. In fact, I'm running a Gordon conference next month, and I plan on using 2 sessions to discuss issues like this. I think things have changed a lot, and what you do with all this data and how you organize it and disseminate it, and how much you can't just, you can Google. I mean, everything is at your fingertips with the internet. But, even if you Googled all day long, you couldn't even come close to finding what you need to do. And what is it? And I think this goes back to the teaching question. What is it? What is the underpinnings we want to teach students nowadays? How has that changed? And I frankly think we don't have enough discussions about stuff like this because everybody's too busy. So we've gone from a stage where we used to have a lot of scientific discussions with colleagues now to the stage over here where everybody's too busy to do anything. So those are the kinds of things I've been thinking about, and I talk about it publicly because I think we have a real problem. But anyhow.
Yeah, interesting. Yeah. Well, maybe there'll be time to, well, you're devoting some time to it already, but maybe there'll be more time, I don't know. But yeah. One thing I wanted to ask you, which is much less serious, but on your website you have a little drawing of a dog on there.
I guess probably, I guess
what's
so.
that? Why? What's that?
I don't know. I think one of the students did that. The website hasn't been updated in a long time because I'm in the dark ages. But that's my dog. Huh. So I have a dog named Enzyme, and so she comes to work with me. She's like 13 years old now. So everybody on campus knows my dog. So I get into the elevator, and no one says hello to me. They look down on the floor and they say, "Hi, Zyme, how are you?" How are you? Her name is Zyme is for enzyme, anyhow. So that's probably why she's on there. And somebody, apparently people send her emails, but I don't pay any attention to that. But she's my best. She pays more attention to group meetings than anybody else because she likes a laser pointer. Yeah. Anyhow, so our department's been pretty good. We have a couple of dogs in our department, and there are a fair number of dogs on campus now. I'm not sure how much longer that's going to last. I think all it takes is 1 or 2 bad experiences, and it will go away, since most places don't allow dogs on campus. But the students certainly like dogs, and I go to, when I teach the lower level courses, they have stubby breaks as opposed to study breaks. And of course, the hit of the stubby break is my dog. Right, because I think they all miss their animals. Yeah. So.
Oh, so you bring the dog to the study break?
Yeah, I mean, she comes to work with me every day, so she's a fixture. And fortunately, she doesn't bark very much. So, otherwise, I don't think she would have been here this long. She would have disturbed people, but she's pretty quiet. So.
Yeah. Well, I love the idea that she's named Enzyme. So.
Yeah. What can I say? It's just another example of my work personality.
Great. Well, let me just see if there's anything else that I thought we might cover, but this has been great. I really appreciate it.
Well, thank you.
And again, if there are other things that any stage of things that we've left out.
No, I mean,
that's
I
important?
don't think so. I mean, I think my biggest thing that would be helpful to young people is they seem to fixate so much on getting tenure and doing this and doing that. I mean, I just sort of think it's too bad they waste so much energy doing that. I see them, and they will submit a paper to 5 places before it gets accepted, right? And in the end, in my opinion, it doesn't really make any difference if it gets peer reviewed in a thoughtful fashion. That's the key thing. But they seem to think that's what they need to do to get tenure. So I don't know. They spend way too much energy on stuff that I think is not important.
But is that what they need to do to get tenure? I mean, is it the system that they're, or is it?
I think it is. I mean, I think I've heard, even within my own department, I've heard, the word scholarly, is not a good word to describe somebody because they didn't publish in this journal or that journal. And I mean, in the end, what's really key is what's in the papers, and does it withstand the test of time. And also, I would say that most people don't ever do anything that's that important. I don't know. So, I mean, we all work and we work. And if I were to ask a student who Frank Westheimer was, who was the founder of modern chemical biology, I would say 99% of the people wouldn't know who he is. So we're just a drop in the bucket. And so they waste way too much time focused on stuff that I don't think is important, instead of just really doing good science and having fun, not spending all this time making sure you get published in Journal X versus Journal Y. So I don't know.
Good science and having fun. That's
having fun. Once you're not having fun, you're in the wrong game. Science is too hard not to have fun. So you're never going to have fun all the time, right? But that discovery is what it is that keeps you going, man. You can't wait to see what the result is. And I think once you lose that, I don't know why you're doing it anymore. Unless you like to eat and drink and travel around the world, which I don't like to do. That's the other part of science, right? But
yeah. Wonderful. Well, thanks so much for your time.
Yeah, okay. Yeah.